We're talking about, we had just brought up sex-linked traits. And these are traits that are found on either the X or the Y chromosomes or the sex chromosomes. Um, uh, they're not always X and Y, depending on what animal you're talking about, but they are in us. Um, Genes on the Y are called Y-linked genes. Those on the X are called X-linked. And I wanted to go through a problem with you so you could kind of see where this is going, because that will be another one of your. Sometimes this computer is like the only thing that this thing doesn't do very well is it doesn't want to change. You are on your essay. One of your genetic problems will be on sex-linked, a sex-linked trait. The X chromosome has many genes on it that are unrelated to sex. In other words, biological sex. So, are you producing ovaries? Are you producing breast tissue? Are you producing hair? Many of the Y genes, however, are related to sex determination. So they tend to be, just sex, biological sex related traits, whereas the X is not. How many times do I have to hit it? It's like the elevator button. Okay. X-linked genes follow a specific pattern of inheritance. And I'm going to go through this. I'm going to say it first, and then I'll go through it with you. For a recessive X-linked trait to be expressed, a female needs to have 2 copies of it. So they need to be homozygous for it. In other words, they need to have 2 copies. Males only need to have 1 copy, and we call that hemizygous. You don't really need to remember that, just because males cannot be homozygous. And I'll explain why that is. X-linked disorders are much more common in males than they are in females because of the fact that most of these disorders, they're X-linked. So because they're X-linked, it's easier for guys to get it. Because let's take a trait of male pattern baldness. Ugh. Oh, all the guys are like, yeah, no bueno. And you can rock the baldness. And what are my sex chromosomes going to be if I'm a biological male? X and Y. And that is a recessive trait. So we're going to say that. Yeah. Baldness means that you have that B allele. So a small B allele. So this guy, hair or no hair? Hair. No hair. Thank you very much. Yeah, thank you. No hair. He's bald. Okay. And he has kids. Let's just go like that so that's not confusing. He has kids with someone who is, let's make her a carrier. Does she have hair? Yeah. She does have hair. Is she a carrier? I just said that. Yeah, she's a carrier. She is heterozygous for baldness. He is just bald. He's recessive for bald. And the way we do these Punnett squares is we have to keep track of not only the trait, but we have to keep track of the X's and Y's and X and X values as well. So when we do that, we female goes across the top, male goes on the bottom. Okay? So I have to say those letters right away. And I know that she is a carrier. But notice how there's nothing on. I didn't put anything here. I didn't put anything on that Y. Because that Y, again, I said that there's very few genes on the Y that are not related to biological sex. and having hair and not having hair is not a biological sex related characteristic. So that Y chromosome doesn't have a bald gene at all. It's just not there. Did I answer the question? So it's not there. Okay, so there is nothing on the Y, so you just leave the Y blank. So now I do the, come back here to my red, and I do.
Dude, there's no G, there's nothing on the Y, so it's just a little B.
I have a big B and I have a small B.
So the male has the recessive ball.
Uh-oh. Hey, everyone's like laugh. All the guys are laughing. Why are you laughing? Why are you laughing? Nate, why are you laughing? I don't know. I don't know why I'm laughing. Okay, so this guy down here, this baby boy, will grow up to have a balding problem when he's older. But what about the female? Oh, she's going to be bald too. Yeah. It's more common in women than you think. Women tend to hide it with wigs, and good wigs can really look like real hair. So, but anyway, it's not a wig. So it's easier for the guy to get it because he only needs one hit. He only needs one allele, recessive allele. The female needs two.
They all have to be though. Are they all bald?
Big B means
You gotta do some more.
Big B means no ball.
You gotta get updated.
He said, I'm gonna ace the test.
No ball. That's just racist.
no baldness. No baldness. They have hair.
No ball.
All right. Got it? All right. So here's my next question. Who did the guy, let's just focus on the guy here, the baby boy. Who did he get his bald trait from? From his mom. From his mom. So, guys, you do not look at your dads for baldness. You look at your mom's brothers. Look at your uncles. Well, then you can't tell.
I'm low-key chilling.
Let's go.
You good? You good?
I'm good. Let's go.
You good?
My mom has
You good?
He low-key like doesn't have that much hair, but he also
All right. All right. Cool. That's sex-linked traits. Why uncles? Because the uncles will have gotten the gene from your mom's parents
But couldn't you go?
that she's not showing, but she could be carrying. So she can be a carrier for it. I mean, you're still not necessarily going to get it, but you could be a carrier. Mom could have been a carrier for it and given it to you. So you look at your uncles, and if uncle's bald, then there's a possibility that you could have it too.
Could you also look at your grandpa from your mom's side?
Wait, you're on my mom's side?
But your grandma on your mom's side also.
You could look at anybody.
Man, I'm not gonna do that. You're not here.
Ouch! You just like
Do I need to?
you just like make fun of his hair.
No, it was a future, it was a prediction. It was just
Dude.
All right. So here's another. Nope. I need to reboot this computer. Here's another example of that. It's just several different scenarios where you have N is in normal, so they're not telling you what the trait is. They're just saying N for normal and small n for not normal. And then these two have children together. And the kind of pinky salmon color here is to represent that they're carriers. And if they're white, they're clear. They don't have the trait. And then showing how, next generation, it could come out. So that person's a carrier, and that female carrier could then pass it on to her male son. Male son? I guess that's redundant. Her male offspring. And of course, the worst or most damaging, I don't even want to say that because it's just doesn't necessarily mean it's a bad thing. But the trait that the phenotypes that would affect them the most would be if you've got a carrier for the mother and someone who has it that's male. Then you're going to end up with both males and females getting it. It's just not easy. Again, not easy because the female has to have 2 copies of it. Okay. So examples of this range from things that are, not a big deal to things that are life-threatening. And I think the next slide talks about things that are life-threatening. Come on. Yeah.
Is there a reason that we become bald?
Is it an advantage?
Yeah, biologically.
I don't think so. No, I don't think there's like an evolutionary, but I think it doesn't hurt you to be bald. It doesn't help you to be bald. So, does it what? Well, I mean, you can think of it in a couple ways. You can think of it, it really is cold to not have hair, right? Anyone who's cut their hair short knows that it's cold, right? I always keep my hair longer in the wintertime because it's so cold. But on the other hand, let's say you're out and trying to survive and you're fighting with somebody else and you have long hair, they can grab it. They can grab it, they can pull it, that could be another way to, could be a disadvantage. That's what I'm trying to say. So, yeah. Color blindness is most types of color blindness are X-linked. Again, so you see biological males getting it more often than biological females. Muscular dystrophy, which is a form of deterioration of the muscles, is another example, as well as hemophilia. Hemophilia is a blood clotting disorder where you don't make one of there are clotting factors. These are proteins whose job it is to make sure that when you cut yourself, your blood clots so you don't bleed out. Well, in hemophilia, one of those clotting factors is missing, and there's several different clotting factors. There's different types of hemophilia. And you don't bleed out from a little cut. Like a paper cut's not going to hurt you. Even a pretty big cut's not going to hurt you because you can always put pressure on that. What will affect you is bruising. So if you fall, say you're falling downstairs, the problem is internal bleeding because you can't stop the internal bleeding as easily. So people who are hemophilic have to be careful about falls. And if they are in, say, a car accident, they may get the clotting factor that they're missing infused into their bloodstream to help with that so that they can clot their blood and not bleed inside. So my computer's just really, really slow today, among other things. Why are you on a finger point there? Come on. Okay. So, now we need to talk about something called X inactivation. This happens in females, biological females. In mammalian females, one of the 2 chromosomes in each cell is randomly inactivated during embryonic development. It's called dosage compensation. And that that one, whichever one is inactivated, it never, for the entire life of that person, it will never become activated again. It's made into a tiny little bundle that is visible under the microscope pretty easily, called a bar body. If a female is heterozygous for a particular gene that's located on the X chromosome, she'll be what you call a mosaic for that character. And I can give you an example of that. Who are my cat people? Who are my cat people? What kind of cat is that?
Oh,
Fluffy cat. It's a fluffy. It's a calico. Yes, it's calico. What flavors of calicos can you have?
chocolates.
What sex are calico cats? Always females, because they're mosaics. Okay. So one of their X chromosomes is inactivated, and if it's inactivated with the black fur gene on it, then that fur will be orange, and if it's the orange gene that's inactivated, the fur will be black. So you get this pattern appearance. Okay. But you only get it in females because the dosage compensation, the inactivation, or the bar body formation doesn't happen in males. So you wouldn't have any. You looked at a slide for bar bodies, you wouldn't find any bar bodies in a biological male that was XY, but you would find one bar body in a female that was XX. Right? Yeah.
You know, like from one eye, like, they're from the other eye. But that can happen in humans. Wait, what?
I don't know, actually. I have to research that one.
Okay.
It's like mixture.
Yeah.
I don't really see it, but yeah, I've seen it. I mean, in dogs, right? You've seen it in dogs. Yeah. I don't think I've ever seen it in humans. I'm not sure if it's a thing.
My sister has the same sort of eye.
Yeah? Really? Okay. I guess it does. What's that?
I always used to look at it.
With what?
Tigers.
Who?
No, I haven't. Oh, I guess there's a name for it. Sophia knows it. Heterochromia. Heterochromia.
What is that?
Yeah.
Oh.
I will look at it after we get done talking and
I didn't know that.
talk more to you about it tomorrow. Heterochromia. All right. So far we've been talking about sex-linked problems. And one of the things that Morgan studied in addition to this monohybrid crosses was he also looked at dihybrid crosses. So he was looking at two inherited characters. And again, he was using fruit flies. And he used a couple of different traits. And they're actually shown in the next couple of slides. Because what he found, I don't know if I should do a spoiler alert or just go through it with you. Man, I'm so sick of this cough. Yeah, let's go. Well, no, I guess we'll start with this. So, small picture. Let me make some of this bigger. Just with, we've got two parents, flies. One who is homozygous for two traits and having the dominant trait, and that is having a gray body. And gray body in this case is, I'm just going to say gray. And having normal wings. That's that guy there. Okay. And crossed it with another fly that was black bodied and had what we call vestigial. That's supposed to be an E there. Can't do my E very well. Vestigial wings. Vestigial meaning they have wings, but they don't work. They can't fly around. So I talked yesterday about how they actually have fruit flies now that you can buy at PetSmart that have vestigial wings. And they do that because people need to feed their critters, their amphibians and their lizards. But they don't want them to fly around the house. So they've genetically modified them so that they can't fly around the house. They can just be eaten by, Wilbur the toad or whatever you happen to be feeding it to. Yeah, Wilbur. All right, so this is the parent generation, and they cross these two. And then, because black and vestigial is recessive, and gray and winged is dominant, you know that all of the offspring are going to be gray and winged. But the question is, what he was looking at was he wanted to see, were they all heterozygous? What were they exactly? He wasn't sure. So he did that test cross. Remember how I said you could do a test cross to see if they're all homozygous dominant or if they're heterozygous? Well, he took the offspring, which were all gray and winged. And now he did a test cross with more of those black and vestigial. So pure homozygous recessive for black and vestigial against gray and winged. And he didn't know exactly if these were homozygous dominant or if they were heterozygous. Okay. So then he, and I say, I'm using the imperial he here. One of the things that graduate students in genetics get stuck with is a lot of fly counting. And so there was some person that was their job, there's actually something they sell called FlyNap, which you put into a tube of the flies, and it puts them to sleep. Doesn't kill them, just puts them to sleep. And then you take them out and you look at them underneath the microscope while they're kind of twitching, because they will twitch, because they're just anesthetized. And then he had to count them. And what I'm going to show you here in the next slide is the results. It's just this, but larger. So that's a couple of slides down.
Wait, so which cross was the dihybrid?
Well, dihybrid is always when you have two traits. So in this case, we're looking at the body color of gray or black and vestigial or winged fruit flies. So there are two traits. And so the predictions were either, so he was suggesting that what might be happening is these genes are on different chromosomes. So if they're on different chromosomes, that whole independent assortment thing is going to play out, meaning that how one homologous pair lines up has nothing to do with another pair lining up. Okay, so independent assortment is in play, and that is a thing. But if the two traits were found on genes close on the same chromosome, then what you should end up seeing is the offspring shouldn't look like anything except the parents. Okay? Because when crossing over happens, if they're on the same chromosome, they tend to break off and be passed over as a package deal to the other chromosome. Remember how I talked about eye color and hair color? Brown eyes, brown hair is usually a thing. Blue eyes, blonde hair is usually a thing, because those are linked genes. They are close together on the same chromosome. It is possible to get someone with blue eyes and brown hair, or brown eyes and blonde hair, but it's not as common because those two genes are close in terms of their distance on the same chromosome. So when he did these two particular traits, what he found, or again, like I said, one of his poor graduate students found, is that he had a large number of flies that looked like their parents. So 965 were gray-bodied and normal-winged, and 944 were black-bodied and vestigial. He did find a few, but not as many, not nearly as many, that showed kind of a hybrid of the two parents. They had the grayness of the one parent, and they had the vestigial of the other parent. And they had the black of one parent and the normal wing of the other parent. Okay? Because it is possible, it's not very likely, but it is possible for two linked genes, when they cross over, to not cross over together.
So your genes. It's like just like medicine.
Here's a chromosome.
It just means I'm one of them.
Like maybe your genes didn't. You were supposed to be blonde, bro. Things like that.
Yeah, that's too easy. I'm gonna need a volunteer.
No, maybe. Max, go, bro.
Do you need to come up there?
No,
Okay.
you volunteering?
Sure.
Okay. You're gonna like this, I think. All right, this is a chromosome. I want you to illustrate crossing over by breaking this chromosome in half. And I would like you to try and break it between those two.
Between the two.
Yeah.
Oh wait, do me though.
What if I don't do it?
It's okay. I prefer you don't, actually.
Oh, I
Oh.
didn't do it.
Okay, it's okay though. That's actually perfect. The point is that so I have a couple chromosomes here.
Round of applause.
Sorry, I have a couple genes here, right? This is my whole chromosome, and they're really close together. So during meiosis, those two chromosomes are gonna cross over. Sorry, genes. Those genes are gonna probably cross over as a unit because they're so close together. Yeah, it can happen, but as Nate says, or as Nate can tell you, it's not that easy. Anybody want a broken pencil? It's always fun. Yeah, so it did happen in 206 cases for this guy, for these guys, for this category, and 185 of them. But most of them are going to be this. And why do I spend so much time on this? I spend time on this because this is one of those questions that they love to bring up on AP exams, where they give you a whole bunch of data. And what we can tell from this data, if those numbers don't come out the way we would normally predict, normally when we do a dihybrid, we assume that there is no independent, one more time, there is no linkage, that there is independent assortment, and that you're just as likely to get gray vestigial as you are black vestigial. These numbers are very different from that. So when you get something that's very different from that predicted values, that's usually a good indication of linkage, that the two genes are linked together. They're very close together. And now that rule of independent assortment doesn't count because they're on the same chromosome. Okay. Very common question that will definitely come up again. So Morgan found that body color and wing size are usually inherited together in specific combinations, those being the parental phenotypes. But occasionally, the genes did not assort independently. Oh, sorry. Not saying what I should be saying. He noted that these genes do not assort independently, and he reasoned that they were on the same chromosome. Now, occasionally, you did see the numbers being different, and that's because that infrequent crossover snapped it in just the right spot to split those guys apart. Okay, so this is the crossing over is just an example of genetic recombination. I'm not sure we've used that word when we talked about crossing over before. But because the crossing over is always different, you end up with unique chromosomes as a result that have different genes on them that you never, in combinations that you may have never seen before. And because of that, you get huge variation. And that's kind of the whole idea with meiosis, is you get a huge variation. Unlinked genes recombine at a rate of 50% due to crossing over. So 50% of the time, if they're not linked together, they're going to cross over. Linked genes can either be on different chromosomes, or they could. The other thing they could be, and that's something to keep in mind, is they can be far apart from each other on the same chromosome. So if I had taken my pencil and, if there was one gene up at the top of the tip and the other one was down by the eraser, yeah, they would cross over all the time. They're on the same chromosome, but they're so far apart from each other that it's like they're on a different chromosome. If genes are close together, the recombination doesn't happen as often. And that will skew the numbers from what you expect them to be with independent assortment. So if you do a cross and the numbers aren't what you expect them to be, that's when we did the dihybrid cross, and I had to do that 9:3 to 3:1. I was assuming independent assortment. I was assuming that those two traits were on different chromosomes. If you get values that are different from the 9:3 to 3:1, in the case of those pea plants, then you know that something's going on, and it's likely that they are linked genes. Alrighty. Morgan observed that although some genes are linked, non-parental allele combinations are still produced. And we didn't, at this point, this was just early 1900s. He didn't actually know, still didn't really know much about chromosomes. And they didn't know about crossing over. So he was the first one to kind of hypothesize that this crossing over was happening based on his results. So he guessed it before we knew about it, before we could actually see it. And as I said before, recombinant chromosomes bring alleles together in different ways in gametes, in the sex cells, that you've never seen them before. So you get this crossing over, you get random fertilization. It leads to a huge number of possibilities of what the genetic makeup will be in each sperm and egg, and therefore in each organism that's produced from that. And that is important because that is what nature's going to work on. So nature's going to work on whatever variation you have, and that's how natural selection works, which we haven't gotten to yet. But I don't know if anyone's like an endangered animal kind of person, but a lot of times they talk about endangered species, and one of the problems with them being endangered is the numbers are so low that there's a limited amount of genetic variation in the population. And just like we talked about, having kids with somebody who you're closely related to, if you've got an endangered species that has a very limited genetic variation, same thing's going to happen. You take two that are not, allegedly related to each other, pheasants or sea lions, and you put them together, even though they are not family tree-wise not related to each other, genetically they may be too close to each other. And that then makes all of their offspring more likely to have different diseases, and it's hard to bring a endangered species like that back because of that reason, because of the genetic variation is usually pretty limited. All right, that seems like a good spot to stop. We're definitely going to get through notes by Friday. My plan is to do a lab on probably Tuesday, and then another activity on Wednesday. And then test will be the following Friday. All right. You should be able to get through